Hi guys, welcome back to my channel. Today I want to talk about G protein coupled receptors mediated signal transduction. Okay, as you can see, this is my plasma membrane, and then outside of my cell, I have this ligand that is waiting to bind to the, as you imagined, the protein receptor that's embedded in the cell membrane, right? And then this receptor has a name called GPCR, and I'll explain what does GPCR mean in just a second. Okay, now let's move on to this green protein. Okay, so this green protein that has 3 subunits is called G protein. Okay, and then G proteins alpha and gamma subunits are attached to the cell membrane. This is called lipid anchor. All right, so in this case, my G protein has 3 subunits, but G protein doesn't necessarily have to have 3 subunits, right? It can also have one single subunit, like the small G protein rest, right? But in case of signal transduction, we want our G protein to have this heterotrimeric structure with 3 subunits. Okay, all right. So now we've gone through 3 components, right? The ligand, the GPCR, and then the G protein, okay? And then because the GPCR is binded to the G protein, so GPCR really stands for G protein coupled receptors, okay? It's not any other receptors, but the G protein coupled receptors. Okay, that's what it stands for. And then also it has 1, 2, 3, 4, 5, 6, 7 domains, so that we also call it 7 transmembrane receptors. But G protein coupled receptor is a more common name. Okay, all right. So now let's move on to GDP. Okay, so what is GDP here? So GDP here is basically a deactivator of G protein, which means when GDP is binded to the G protein, the G protein is inactive. Okay, so this whole thing is inactive now. Okay, if we want the G protein to be active, it has to bind to GTP instead of GDP, right? And then how does that happen? It can simply done by the binding of ligand with my GPCR. Okay, so now let's bind them. Okay, so now this other picture you see my ligand in this case is already binded to my GPCR, and then after the binding you see this conformational change of GPCR. Okay, and then due to the conformational change, this will disassociate beta gamma units from the alpha unit. Okay, and then also you see now my alpha subunit is attached to GTP now instead of a GDP. Okay, so after the alpha subunit is activated, by saying activated, I mean it's now binded to GTP. Okay, so now it's binded to GTP, it's activated, it will bind to another effector enzyme. Okay, this yellow thing is effector. So you see this effector, when everything was inactive, this effector just hangs in the cell, right? But now the alpha subunit of the G protein is activated, so that my effector enzyme can bind to the alpha subunit, and this causes more downstream biological cascade events that leads to the final biological effects that we want. Okay, all right, so this is a big picture. Okay, this is the picture how G protein coupled receptor can mediate signal transduction, right? The ligand is the very first signal that comes from outside of the cell, and then it binds to the surface of the cell, if you will, and then it will cause the downstream cascade biological events inside the cell. Okay, all right. So now I'm gonna give you 2 very specific examples that has their specific ligands, their specific effector enzymes, and their specific biological effects. Okay, so the first one is when you back in the days when you learned the nervous system, remember the sympathetic nervous system where there's this fight or flight response, right? So this is actually due to the binding of epinephrine, in other words, adrenaline. So epinephrine in this case is the ligand. It's a neurotransmitter, right? This ligand binds to the GPCR. Okay, so once it binds to GPCR, once again GPCR undergoes conformational change. Okay, and then that eventually activates the alpha subunit of G protein, and then it will bind to the effector enzyme. Okay, and then the effector enzyme that gets bind is called adenylate cyclase. Okay, so this is the effector enzyme that binds to the alpha subunit, okay? So this will cause the turning of ATP into cAMP, right? cAMP is the cyclic adenosine monophosphate, right? Okay, so this is the downstream effect that I'm talking about, right? From this point on, we're having this downstream biological effect, okay? And then cAMP is really the second messenger, okay? And then the first messenger is the ligand, the neurotransmitter epinephrine, right? In this case. Okay. So the second messenger can really activate other biomolecules, right? In this case, it's pKa, okay? Which is not a pKa that you learned from chemistry class, but it is the protein kinase A, the pKa in the biological world, okay? All right, so this kinase can phosphate whatever molecule, right? And then which will cause the final result of, let's say, the dilation of your blood vessels, right? So this kind of responses, this kind of gives the fight response, okay? All right, so dilation of the vessel is just one of the biological effects that I mentioned, but it can really be a lot of the biological effects that it causes, right? And then together they facilitate the fight response, okay? All right, so this is one pathway, right? So another example is when you learned the muscular skeleton system, there's a neurotransmitter called acetylcholine. Okay, so acetylcholine is the ligand now, okay? It's the ligand that binds to the G protein coupled receptor. And then that causes the conformational change of the receptor. Eventually, it activates the G protein, the alpha subunit of the G protein, right? And it will bind to the a different effector enzyme, which is called phospholipase C. Okay, so in this case, my effector enzyme, this yellow thing, effector enzyme is phospholipase C, okay? So what phospholipase C is, it would facilitate breaking down PIP2, okay? And then the PIP2 will be broken down into two parts, and one being IP3 and then one being DAG, okay? So in this case, the second messengers are IP3 and DAG, and then one more on the way, okay? So the other one on the way is IP3 will bind to, will kind of activate the calcium transporter on the ER, okay? So that releases the calcium concentration, release it from inside of the ER to the cytosol, okay? And then together, calcium and the DAG, they're gonna together activate PKC, which is protein kinase C, right? So here, my second messenger, I have three, IP3, DAG, and calcium, okay? And then cAMP is the second messenger in my first case. Okay, so what does PKC do? Then PKC, yeah, it's a kinase. It will phosphate whatever molecule, right? Eventually, it can reduce, let's say, comodulin binding, reduced muscle contraction, right? Okay, so muscle contraction, once again, this is just one of the biological effects. It's just an example, but really, like, this is more complicated than that, right? This can, I mean, for the protein kinase C can really also activate glucagon synthesis, let's say, okay? So I don't want you to like memorize everything I said today. I want you to get the gist that protein-coupled receptor-mediated signal transduction is very widely seen in the biological system, okay? So it starts from extracellular signal, which is a ligand, and then it binds to the famous G protein-coupled receptor, okay? And then which eventually it activates my G protein, which activates my effector enzyme, and then all the downstream biological activities can happen, right? So once you know this big picture, once you have this this structure, this logic flow, then no matter what MCAT throws on you, if MCAT comes up with let's say a super complex, super difficult, like a ligand that you've never seen, it's okay, right? It's just the name of the ligand, but you know the mechanism. You know the ligand binds to the G protein coupled receptor, and you know what happens after. So it doesn't matter. It throws you, a different effector enzyme, a different second messenger, or different biological effects eventually. It's okay. You just follow the passage. You read the pathway. You get an idea who is the ligand, who is my effector, who is my second messenger, what is my final biological effect. Then you're gold, okay? So as complicated or intimidating the chemistry or physiology can be in terms of a human body, you can really extract lots of the patterns or similar logic structure for different contents, if that makes sense, right? So the takeaway is really the pattern that we come up with, right? And then also just to show you this table that I came up with when I first learned this, right? So yeah, I learned the pathway. So I want to fill my path. I learned the general pathway. I want to fill the general pathway. So my two examples: what is my ligand? What is my effector enzyme? What is my second messenger? And then how do they lead to different biological effects, right? So it's really just a structure, right? The pattern that then you're not scared of anything. When you practice medicine, right? It does take, critical thinking and reasoning to fit each unique scenario into the big template-like picture. But you want to have the picture. You want to have the big structure first, okay? Yeah, and then I want to share my 2 cents regarding the study of medicine. This fitting each scenario into the bigger template picture is a very fun thing to do. But it's also really fun to just think outside of the box and embrace all the exceptions, randomnesses that we never encountered before, right? So it's really super academically and intellectually rewarding, but more on top of that is the part where you get to apply all these knowledges and embed all you have learned into human reactions, right? Human to human communications, by which I mean treating your patients in the future, right? So medicine is really also a test of emotional intelligence and also a practice and polish of your personal characteristics like empathy, sympathy, and all the other great attributes, right? So isn't that beautiful to have all of these? The combination of lifelong academic pursuit and the caring for human communications, right? We learn from our patients. The combination of all this, do this for the rest of your life. Isn't that beautiful and exciting? Sure thing, right? The answer is 120% positive, okay? So, all right, so enough of my 2 cents. But if you like my videos that promote medical education and communication, and if you feel my passion and love for this, please subscribe, like, or share. And I wish you the best luck with the rest of your journey pursuing medicine. I hope that's helpful.